there has been a significant reduction in cost over the timeline of the Human Genome Project. And the top line there is a $3 billion price tag. It wasn't just the sequencing cost of the human genome, but this is the entire check that the governments around the world received in sequencing a number of model organisms. And as you just go down the line, you'll see that the costs have decreased. And really the dividing line is between 2007 and 2008, where the Watson genome and the Venter genome were sequenced by two different technologies. Venter by standard Sanger sequencing, and it's less expensive than the Human Genome Project, but still a quite hefty price tag. There's a second type of template preparation, which is called single molecule. And single molecule can be accomplished in three different ways. You have a template, you have a primer, and you have a polymerase. And you can attach any of those to a surface and then bind the other parts to generate an active polymerase species. Helicose uses two of those three methods. They first came out with a primer attached to the surface. You hybridize a template, add a polymerase. They've also come out with a strategy where you can attach the template to the surface, hybridize a primer, add a polymerase. A lot of this should, credit and progress should go to the NHGRI. They had a roadmap in 2003 and really understood that we needed something different if we wanted to sequence more than one genome, that Sanger sequencing was just too expensive, too time-consuming, too laborious. And they've put in more than $100 million into next-generation sequencing technologies, and many of the platforms have had NHGRI funding to get them where they are today. This is a changing field. This is not static at all. The technologies continue to get better, new players enter the market, and there are still a list of 6 or more companies that are all trying to get new technologies into the market. Currently, the biggest challenge in next-generation sequencing technology is handling all the data that's being generated and trying to make sense of it. And there's really more on the horizon. And I really see the future in single molecule. I think more and more technologies will be geared towards single molecule.
For surveying the allele-specific expression across the genome, we used bead arrays, which were called NS12 bead arrays from Illumina. And these bead arrays are enriched for SNPs in the coding regions of the genes, so we thought that they were quite suitable for this type of analysis. So this array contained about 12,000 SNPs in 8,000 genes. And you can see by the blue bars here in the picture, the blue ones are the ones that show where we have SNPs included on the array, and then the light blue ones show that, and then the darker blue one shows where we have heterozygote SNPs, which are informative for this expression analysis. Then we used the data, the methylation data then from from 400 ALL patients, the methylation sites in 400 genes in about 400 ALL patients to do this type of classification analysis. And in this nearest shrunken centroid classifiers, what you do is that you try to identify the smallest number of genes that gives a reliable distinction between the ALL samples of which we knew what subtype they are. We can see that we need 9 CPG sites to make a very robust distinction between B cell and T cell ALL, which is perhaps not so surprising, and these cells can be distinguished quite easily by other methods also. But then also for the subtype, the 4 largest subtype groups of this BCP ALL, we also, using about 40 CPG sites, we could make a robust classification of the ALL samples according to the cytogenetic phenotype.
Why Dagestan? Dagestan is not as famous as Tibetan and Andean population for high altitude adaptation, but still there are people who have been shown through genetic and archaeological evidences of being living at 2,000 meters for at least 10,000 years. They are endogamous population and they have a reduced gene flow and strong genetic drift. In fact, on this MDS plot calculated on Y chromosome upper groups, we can see that a Dagestani cluster is clearly individuable among population belonging to the J1 upper group. The derived allele for this NIP might have been lost from the lax gene pool to prevent hypoxic stress causing HF1 related diseases, and that the cluster of intronic SNPs observed in Kubakians for EGLN1 might reflect adaptation to high altitude. And also, I was happy to find that this same gene was found under selection in another Tibetan studies by Simonson et al. 2010.
We have to solve these details of being able to make the variant data that we know is out there accessible as soon as we can. We know that when data is subject to access controls, it's a burden to use, and it does limit the number of researchers that will reuse the data. The HapMap data has been reused by thousands of researchers, tens of thousands almost certainly. Something like 1,100 have applied for access to the Wellcome Trust Case Control Consortium data sets, although they're incredibly valuable resources. Still, it's necessary, and so the policy and technology in this area are important. It's also important to remember that 5 years ago, accessing this type of data wasn't just difficult, it was impossible. And so to make it just really difficult is progress. And we need to make it continually less difficult over time. And it's important as well. So this was a commentary in Nature Medicine over the summer. It's important as well to keep in mind that the public is watching this. So this commentary has a headline, "The delay in sharing research data is costing lives." Now, in and of itself, this is quite an inflammatory headline. But if you read into the commentary, the statement that is made is, and I pulled it out, I'll read it here, is, "Making science work fast enough for patients will require researchers to treat information with greater urgency. Surely, if anyone knew that he or she possessed life-saving data, he or she would act swiftly to share it, just as an intelligence officer would rush to report evidence of an impending terrorist attack." When failing to share data becomes equivalent with preventing terrorism, there's a change taking place in the public thinking about how we produce and share research data that can impact human health in society. And so I think we have to keep our eyes on this. For those people who believe that data should be widely shared, in the end, this is a laudable goal. Methods such as this and these statements, they might be slightly more extreme than needed to get the goal done.
Initially, people think that not individual bacteria, but community very important to know, because some of them responsible for chronic disease, and also some bacteria cannot be cultivated, and you can use only DNA sample from environment with direct sequencing. And this sample will produce short sequences, high frequency of error, and it will be pretty heterogenic, and therefore it puts some challenges for computational gene finding. And I participated in the first published work where about sequencing of bacterial community, and people took a sample from environment, and we developed a pipeline specifically to annotate this community bacterial sequences. Currently, we try to use this pipeline for diagnostic of infections and environment and food contamination. This task is important because, for example, in hospital infections, if you identify what bacteria in a sample, you can save a lot of lives. The same for if you would know bacterial population in wounds, gut, and in, of course, like biological terroristic attack might be. So to identify what particular bacteria species in a sample, we try to apply next generation sequencing data and our pipelines.